So my name is Liz, and this summer I have the pleasure of working on Andrews Nelson Lab. I was under the mentorship of Kaylin Rayleigh, and we were studying of the impact of link RNA on seedling development. So I'll start explaining what is a link RNA. It stands for long intergenic non-coding RNAs, which are RNA transcripts that do not encode proteins, and but have an important role in transcriptional regulation of other genes. We were looking specifically at the link RNA germination defects 1 or LGD1, and this is a locus that harbors multiple long non-coding RNAs. And these are thought to be linked to germination defects in Arabidopsis thaliana when you disrupt it. So we look at LGD1 and its position in the genome, and we notice that it's close to R2A. R2A is a protein coding gene that is related to the recycle of carbohydrates during the seedling development. So our hypothesis was, if we disrupt LGD1, will be this impacting R2A expression, and furthermore will impact seedling development. To test this, we collect mutant lines, we sow them in different treatment medias with MS and MS plus sucrose to check sucrose as our carbohydrate source. We measured seedlings area from day 1 and day 7, and we did RNA-seq analysis on seedling tissue to determine differential abundance. Our results in MS and MS plus sucrose didn't show any difference of growth, and we also checked for each treatment that we did, and there wasn't any difference of the seedling area growth, but we still observed interesting results in the RNA-seq analysis, and we observed that mutant lines are expressing differently to wild-type lines, telling us that LGD1 didn't affect R2A expression, as I mentioned here. So here, this heat map is telling us that these are our mutant lines, and this is wild-type lines, and you can see there is the same shade of color, meaning that there is expression of R2A. Then LGD1 didn't affect it at all. But we wonder, okay, is LGD1 affecting on any other genes? And we did the studies of upregulated genes and downregulated genes. We observed that downregulated genes are being impacted in certain way, and these are related to biological processes in cellular response to hypoxia and jasmonic acid signaling pathway. So for the future, we have to check if LGD1 is affecting any of this biological process, and also if it has any impact in maybe long distance genes in the genome. Yeah, thank you.
Very nice. Thank you. What was your favorite part of this project?
Oh, I love learning about RNA-seq analysis. I didn't have any background in bioinformatics, but I learned a lot, and making these graphs was so much fun too.
Great. Well, thank you for sharing your work with us.
Thank you so much.